Alright, so the last two lectures have discussed the mechanisms, both cellular and molecular mechanisms that underlie hypertrophy in response to different training methods. Now we're going to go ahead and hone in on two parts, which focus on variables which can maximize hypertrophy. So this is part one of that series. One of the first mechanisms we think that is responsible for hypertrophy that's induced via training is metabolic stress. So basically, metabolic stress originates from acute depletion and restoring energy, or ATP, by anaerobic energy metabolism. The purpose is essentially to minimize cellular disturbances, and it's associated with change in calcium homeostasis, induced possibly by hypoxia and ischemia. We generally see an increase in ADP, AMP, inorganic phosphate, pH goes down, glycogen goes down, heat goes up, and we get an increase in osmotic stress, cell swelling, an increase in myokines, hormones, and growth factors. So it's pretty complex metabolic stress. It's not just lactate. Mechanical stress is another way we think muscle grows. And this essentially originates from forces that disturb/deform the integrity of skeletal muscle. It causes essentially mechanically or mechanochemically transduced molecular and cellular responses in the myofibers. And as I discussed previously, this may be mediated by phosphatidic acid or IGF-1 or other growth factors like hepatocyte growth factor. All right, so let's talk about like different ways that we can induce these. Can you actually sort of tease out metabolic versus mechanical stress? Because oftentimes they're developed together. Well, in extreme cases, we can use high load, eccentric exercise with fast speed. So you have maximal metabolic stress, but low metabolic stress. In those situations, we see clear hypertrophy. Also, we can have blood flow restriction exercise with low loads, where you have maximal metabolic stress with low mechanical stress. As we talked about before, we basically have that resistance exercise itself is a very complex process as far as the metabolic sort of changes that occur. So, what's the evidence for mTOR being involved? Well, we discussed this before, but here we're looking at a long-term resistance training program, and you're looking at mTOR signaling on the vertical axes of each of these graphs. You see that mTOR signaling is related to change in 1RM squat strength, fat-free mass of the leg, fat-free mass of the whole body, and type 2 cross-sectional area. This was a tremendous study where they looked at metabolic stress following 4 weeks of blood flow restriction training. So what you're seeing is, to the left, basically how much pH decreases from rest, which is equal to an increase in acidity. And on the horizontal axis, you have cross-sectional area. You see that as pH decreases during exercise, cross-sectional area increases. So it's supporting the fact that metabolic stress is important for growth. We also see when inorganic phosphate increases on your graph to the right, that is essentially predictive of growth as well. And we can also see here that things like an increase in ADP from rest is predictive of increase in myofiber size and 1RM strength, as well as other metabolites like H2PO4. Now, why is this the case? Why is this the case? Well, a good friend of mine, David Gunderman, took cells, muscle cells, and put them in culture. Here you're looking at P70 and ribosomal protein S6 and 4-ABP1, all regulators of protein synthesis via the mTOR pathway. What you're basically seeing here is that the control group, which is the clear group, the open bar, and the lactate group, which is the closed black bar, lactate increases mTOR signaling in all of those three groups. Here's what we think happens. Basically, an early stage in glycolysis is a production of glucose 6-phosphate. You remember that's one of the first things that actually occurs during these types of events. Now, basically the addition of a phosphate group on glucose, what that does, it traps glucose in the cell. It then proceeds to be converted to glucose 6-phosphate, which is then acted on by the rate-limiting enzyme phosphofructokinase. Lactate seems to work by inhibiting the rate-limiting enzyme in glycolysis known as phosphofructokinase. So what does this mean? The enzymes basically, if the enzyme is inhibited, there's a buildup of glucose 6-phosphate. And glucose 6-phosphate stimulates protein synthesis. So again, if I were to shorten this up, lactate inhibits essentially phosphofructokinase. So you basically have a backup of glucose 6-phosphate. And glucose 6-phosphate seems to actually trigger a protein synthesis response. So if you look at Gunderman's data here, we're looking at basal levels of high intensity training versus blood flow restriction. And then you look at OH, which was after, and you see that glucose 6-phosphate is higher in the black or blood flow restriction group than the high intensity group. So, that's probably one of the mechanisms mediating this response. Now, so how do we optimize that metabolic stress besides blood flow restriction? Häkkinen at Jyväskylä did a study where they did 10 sets of 10 RM with 1 minute rest versus 20 sets of 1 RM. So the 10 by 10 ended out resulting in a huge increase in blood lactate levels. So we see that to the right, whereas the 20 sets of 1, which would have low metabolic stress but high mechanical, had less. Actually, it had almost no metabolic stress. So again, basically at a higher repetition range, sets of 10, 1 minute rest, you maximize metabolic stress. So what about rest period lengths? We know that sets of 10 might be better than sets of 1. So Kramer did a classic study here. He had, to your right, primary strength workout, where you basically have sets of 5 repetitions with 1 minute rest versus sets of 10 repetitions with 3 minutes rest. And then we also had sets of 10 with 3 minutes rest and sets of 5 with 1 minute rest. So 2 groups with 5, 1 and 3 minutes rest. 2 groups of 10, 1 and 3 minutes rest. Remember, 10 is the ideal number for metabolic stress, but what's the ideal rest length? If you look here, the 10 sets of 10, which is optimal for metabolic stress, with 1 minute rest. If you look at blood lactate pre to mid to even 5 minutes post, it's drastically elevated. If you look at 10 reps with 3 minutes rest, blood lactate doesn't rise. So basically, you're primarily relying on glycolysis during sets of 10, but the rest period length, that when it's long, allows lactate to be cleared. So metabolic stress is low. So how long can you rest to still get the metabolic benefits? Here they're looking at baseline versus post training, and they have 60 minutes rest, which is P60, 90 seconds rest, and 120. All 3 groups had high metabolic stress. So what we suggest is that about 30 to 120 seconds rest should get good metabolic stress responses. But past that, and that will be diminishing. So what does this mean? Likely for movements like leg extensions and leg curls, which are isolation, you can use 30 to 60 seconds rest. But for more compound movements like squats where you can get winded easily, maybe 90 to 120 seconds rest for metabolic training. So here what you're looking at again in support of Häkkinen's study, they had hypertrophy was 8 to 12 reps, which is the H, and you're looking at blood lactate levels, strength 1 to 5 reps. You see moderate metabolic stress, and then power 40 to 60% 1 RM, and then rest conditions. So metabolic stress again is optimal when you're training in an 8 to 12 rep range, and you have short rest period lengths. Last thing I want to talk about is the cell swelling theory. Basically, it's been proposed that, and this is from Brad Schoenfeld here, it's been proposed that increased pressure against the cytoskeleton and or cells perceived as a threat to cellular integrity, which causes the cell to initiate a signaling response that ultimately leads to reinforcement of its ultrastructure. In other words, cell swelling causes the cell to grow. We see that here. When cell swelling increases by the glycolytic system, you get an increase in intracellular fluid, as opposed to when you have a lot of rest between sets, like the aerobic condition. So again, it's got to be metabolic. It's got to be stressing the glycolytic system, which is going to be more 8 to 12 reps. Why the glycolytic system? Well, if you look here, if you stress the glycolytic system, you're going to build up a lot of metabolites, like lactate, like inorganic phosphate, like ADP, like AMP. Metabolites have an osmotic pressure. You draw water into the cell. That senses it as it's a volume sensor. That triggers mTOR signaling, MAPK, which alters gene expression, as well as protein synthesis. So here's Jacob Orms here, who's jacked. And you see how full his muscles are. Well, part of that is his bodybuilding. You know, bodybuilders have 50% more intramuscular glycogen than sedentary controls. And it's likely because they're training in these metabolic ranges. So let's summarize the impact of metabolic stress here. You have metabolic stress by doing 8 to 12 reps, supersets, giant sets, short rest periods, 30 to 120 seconds. So you have a metabolic buildup or blood flow restriction. That activates mTOR. It also activates MAP kinase pathways, which leads to an increase in muscle protein synthesis. We also know metabolic stress may lower myostatin, as indicated by blood flow restriction data, and that activates satellite cells. Thank you very much, and I'll see you next lecture.